Welcome to The Explainer. Today we are going to zoom way in, like microscopically in, to look at the choreography that makes every living thing, and yet that includes you, genetically one of a kind.
I mean, you get your dad's eyes, maybe your mom's smile, but you're not a clone, right? Well, the answer lies in this incredible cellular dance called meiosis. And today we're going to zero in on its most critical opening act, Prophase-1.
Whoa, okay. I know what you're thinking. That looks like a bunch of complicated science words. But don't worry, you don't have to memorize any of this. Think of it as our roadmap. We're going to walk through these 5 stages, and I promise we'll focus on the incredible story of what's happening, not just the jargon.
All right, so every great performance, every great dance starts with the dancers getting ready and finding their partners, right? Well, it's pretty much the same thing inside our cells. This is what happens in the first 2 stages.
So before the show can really begin, all that genetic material, which is usually just floating around like a tangled mess inside the cell's nucleus, it's got to get organized. It has to get ready for its moment in the spotlight.
And this is that first step in action. So just imagine a huge tangled up ball of yarn. That's basically the cell's DNA most of the time. In this stage, the cell starts to carefully untangle and wind up each strand into these nice, neat, distinct chromosomes. And you'll see they start moving toward the centrosomes. You can think of those as the choreographers of the show, heading to opposite ends of the stage to direct everything.
Okay, so our dancers are ready. Now it's time for them to find their partners on the dance floor. And this is a really crucial step. This is where the chromosome you got from your mom finds its perfect match, the one you got from your dad.
And check this out. This is not just a casual how do you do. A special protein structure, the synaptonemal complex, literally zips these 2 chromosomes together all the way down their entire length. It's a super precise, super tight embrace. And this fully paired up structure, we call it a bivalent, is now locked and loaded for the main event. All right, the chromosomes are paired up, and now we get to the absolute heart of this whole thing. This next stage is the entire reason this dance is happening. It's the moment that creates almost all genetic diversity. This is the climax of our story.
Yeah, I mean, everything we've talked about up to this point, all the untangling, the pairing up, the zipping together, it's all been the warm-up act. It's all been in preparation for this one single game-changing event.
So because the partners are held in that super tight embrace, they're in the absolute perfect position to trade a little bit of information. This exchange has a name. It's called crossing over, and it creates brand new combinations of genes, combinations that have never existed before.
Think of it like 2 dancers who are so in sync that they can flawlessly swap a piece of their costume right in the middle of a performance. It's an incredibly precise cut-and-paste job, all run by a whole team of special proteins. They are literally shuffling the genetic deck. And this is the engine of evolution. This constant shuffling is what creates new possibilities, letting species adapt and survive. It's why a single disease doesn't just wipe everyone out, because some of us, just by pure chance, will have a new combination of genes that makes us resistant.
Okay, so after that amazing genetic swap, our newly unique chromosome pairs, they have to get ready to go their separate ways. The dance is kind of winding down now, and the cell is getting ready for the big split.
And you can really see how their connection changes here. That full-length zipper that was holding them so tightly together, it dissolves. But they don't just fly apart. Nope, they stay physically linked, but only at the exact spots where they swapped their genetic secrets.
And these connection points actually have a name, chiasmata. You can literally see them under a microscope. They look like little X's. They're the physical proof that the crossover happened. It's like a lasting handshake, holding the partners together just a little bit longer before they have to say goodbye. And that brings us to the grand finale of Prophase-1, diakinesis. This is basically the last bit of housekeeping. Think of it like the stage crew coming out to clear the floor for the next big act, which is where the chromosomes are going to line up in the middle of the cell before they get pulled apart.
So here's what happens. Those connection points, the chiasmata, they slide all the way to the very tips of the chromosomes. The chromosomes themselves coil up super tight, becoming really short and dense. And then, poof! The barrier around them, the nuclear envelope, just dissolves. The stage is now officially clear, and the cell is totally ready for the next phase of division.
Okay, so after all of that, the pairing, the zipping, the swapping, the preparing to separate, what was the point? What did this incredibly complex dance actually achieve? Let's zoom out for a second and look at the big picture.
Here's the takeaway. The chromosomes that finish this dance are not the same chromosomes that started it. Not at all. Each one is now a totally unique mosaic, a patchwork quilt of genetic info from both of your parents. And that is exactly why you are a one-of-a-kind combination of them, and not just a simple copy of one or the other. So making sure this process happens flawlessly is, well, it's absolutely vital for healthy life and for the diversity that keeps us all going. Which, leaves us with one last really fascinating question to think about. Given just how complex this all is, what happens when this beautiful, intricate dance makes a mistake?